Imagine diving 30 meters beneath the waters off the coast of Indonesia. In front of you stands a brilliant pink Gorgonian fan coral, a delicate underwater tree covered with hundreds of tiny polyps no larger than pinheads. You take a photograph, finish your dive, return home, and upload the images to your computer. Then, while zooming in on one of the shots, you notice something strange. An eye, then another. And suddenly, an entire creature emerges from the background, a tiny seahorse no larger than your fingernail, perfectly camouflaged against the coral. It had been there the whole time, right in front of you. You were looking directly at it and never saw it. This is Bargiban's dwarf seahorse, a species barely 2 centimeters long. It was discovered in 1969, completely by accident, when researchers collected a Gorgonian coral and brought it to the surface for an aquarium display. As they examined it, one scientist noticed something impossible. Part of the coral appeared to move. What they had found was one of nature's most astonishing examples of camouflage, an animal so perfectly adapted to its environment that it becomes nearly invisible. But that's not the story we're here to tell. Because hidden inside this tiny seahorse is a clue to one of the deepest mysteries in all of physics. So what is the meaning of life? You might answer that it is reproduction, the process of passing your genes into the future. This is one of nature's fundamental principles. Every living organism strives to create copies of itself, allowing its genetic information to survive through natural selection. And when we talk about copying in biology, we usually mean reproduction itself, the creation of offspring that resemble their parents. DNA is passed on, traits are inherited, and evolution takes its course. But Bargiban's dwarf seahorse performs a very different kind of copying, and it is far stranger. Its skin is covered with tiny bumps called tubercles. In shape, size, color, and arrangement, these structures perfectly mimic the coral polyps among which the seahorse lives. A gray-purple seahorse inhabits a gray-purple Gorgonian. A yellow-orange seahorse lives on a yellow-orange one. Every bump on its body appears to be a miniature copy of a coral polyp. But wait a second. A coral polyp belongs to an entirely different species. In fact, it comes from a completely different branch of the tree of life. Between the seahorse and the polyp lie roughly 600 million years of separate evolutionary history. And yet, over the last 18 million years, this tiny seahorse evolved the ability to create structures on its own body that are virtually indistinguishable from the organs of another organism. This is not DNA replication. There is no coral polyp gene hidden within the seahorse genome. Instead, the seahorse possesses genes that control skin, growth, developmental pathways, and cellular organization. Under the right evolutionary pressures, those genes produce structures that, from the outside, become nearly identical to the polyps of an entirely different species. And when replication reaches this level of precision, we begin to move beyond biology alone. We enter the realm of something deeper, the realm of physics itself. In 2025, scientists published a study that attracted significant attention. They sequenced the genome of Bargiban's dwarf seahorse and compared it with the genomes of other seahorse species. What they discovered was remarkable. Roughly 18 million years ago, this species split from its closest relatives. Since then, it has completely lost 438 genes. Another 635 genes have deteriorated to the point where they no longer function. And more than 5,100 additional genes have lost parts of their regulatory networks. Do you see the mystery? Most popular stories about evolution focus on increasing complexity. A simple cell becomes a complex cell. Single-celled organisms become multicellular life. Nervous systems emerge. Brains appear. The story is usually told as an endless climb toward greater sophistication. But Bargibanti's dwarf seahorse followed a different path. It became simpler than its ancestors. It reduced parts of its immune defenses because the coral it inhabits is already protected by powerful toxins that kill many potential predators. Why spend precious energy maintaining expensive biological systems when you live inside a fortress built by another organism? Its snout became shorter because resembling a coral polyp proved more advantageous than behaving like a traditional predator. And over time, it lost the ability to thrive anywhere, except on a single type of coral. This is not degeneration. It is not failure. It is optimization in its purest form. Biologists have repeatedly attempted to breed Bargibanti's dwarf seahorse in captivity. So far, they have failed. The seahorses die, the corals die. Even under the care of highly experienced specialists, the entire system eventually collapses. Think about that for a moment. We clone mammals. We grow tissues and organs from stem cells. We edit genomes with molecular precision. We have sent autonomous robots to Mars capable of choosing which rocks to drill without human intervention. Yet we cannot reliably maintain a 2-centimeter seahorse inside a glass aquarium. What does that tell us? Perhaps Hippocampus bargibanti is not truly a separate organism at all. Perhaps it is merely one node within a much larger system. The seahorse, the gorgonian coral, the flow of plankton, the temperature of the water, microbial communities, predators, and the rhythms of the currents flowing through the coral triangle may all be parts of a single physical entity. And perhaps the reason we cannot reproduce it is because we still do not know which of the countless interacting variables are essential and which are not. Remove any component from this system, and the entire structure begins to fail. If the coral dies, the seahorse dies. If the surrounding ecosystem changes, the connection unravels. And this brings us to the deeper idea, whose outlines we are only now beginning to see. In 1976, Richard Dawkins published a book called The Selfish Gene. It permanently changed the way we think about life. Before its publication, most people viewed the living world in roughly the same way. Animals struggled to survive so they can continue their lineage and preserve their species. A lion chases an antelope at full speed, risking broken bones with every stride. The antelope runs with everything it has to avoid becoming dinner. A salmon swims thousands of kilometers upstream, battering its body against rocks to reach its spawning grounds, lay its eggs, and die. Every one of these creatures appears driven by the same objective: survive, reproduce, and continue the line. The organism seems to be the central character of the story, a living being fighting to remain alive. Dawkins turned that picture upside down. He argued that if you look closely enough, neither the lion nor the antelope is the true protagonist. The real protagonist is the gene, a tiny fragment of DNA, a piece of information capable of making copies of itself. Viewed from that perspective, the entire story of life changes. A gene cannot do much on its own. It is merely a sequence of chemical instructions. But if those instructions can construct a body around themselves, a machine made of muscles, bones, nerves, and senses, a machine capable of finding food, escaping danger, and reproducing, then the gene can create copies of itself. Those copies then build new machines, and the process repeats itself for billions of years. Dawkins called these bodies survival machines. A lion is a machine built by lion genes to help produce more lion genes. A deer is a machine built by deer genes, and so are you. Your body is an extraordinarily sophisticated biological system assembled by your genes to accomplish one primary task: survive long enough to pass those genes on. From the gene's perspective, once that task is complete, the body has fulfilled its purpose. It sounds cold, almost unsettling. Yet this seemingly ruthless idea suddenly explained behaviors that had puzzled biologists for generations. Consider a honeybee. A worker bee stings an intruder and dies. The act tears part of her body apart. If survival is the ultimate goal, this behavior makes no sense. But if the goal is the preservation of genetic copies, everything changes. The bee's sisters share many of the same genes. By sacrificing herself to protect the hive, she helps ensure the survival of thousands of copies of her own genetic information. The bee dies, but the genes continue. Or consider the cuckoo. It lays its eggs in the nests of other birds, tricking them into raising its offspring. From the perspective of what is best for the species, this seems absurd. From the perspective of the selfish gene, it is a masterstroke. The cuckoo's genes have effectively recruited another animal's time, energy, and parental care for their own replication. Even some of the most powerful human emotions can be viewed through the same lens. The fierce love parents feel for their children, a mother's willingness to risk her own life to save her child. Affection, tenderness, devotion. Dawkins argued that these behaviors emerge because children carry copies of their parents' genes. Genes that build organisms inclined to protect their offspring are more likely to persist than genes that do not. Over millions of years, natural selection transforms that simple logic into what we experience as parental love. That is the extraordinary power of the idea. One simple rule: that genes which replicate more effectively become more common, can explain sacrifice, cooperation, deception, and love. No grand design is required, no guiding intelligence, only replication and selection, repeated across immense spans of time. Dawkins demonstrated that much of the complexity of the living world can emerge from this single principle. And that is why the selfish gene became one of the foundational concepts of modern biology. Ask a contemporary biologist what life is, and many would offer some variation of the same answer. Life is a system that stores information about itself, copies that information, and passes it forward through natural selection. Genes are units of information. The replication of genes drives evolution. And everything else is, in one way or another, a consequence of that process. Now, let's apply that same line of reasoning to the dwarf seahorse. If the selfish gene truly explains all of life, then it should explain this animal as well. Let's begin with its most remarkable feature: the bumps covering its skin. According to the gene-centered view, the explanation seems straightforward. Somewhere in the seahorse genome, there should be a gene, or perhaps a collection of genes, that instructs the body to grow these structures in exactly the right places, with exactly the right shape and color. Any ancestor carrying such a trait would have been better hidden from predators, more likely to survive, and more likely to reproduce. The genes responsible for camouflage would spread through the population. Classic natural selection. A gene that improves concealment replicates more successfully than one that does not. At first glance, the story seems complete. But when scientists analyzed the dwarf seahorse genome, they uncovered something far more surprising. They did not find evidence that these structures appeared because the animal gained new biological instructions. Instead, many of the changes appeared to involve the loss, silencing, or disruption of existing developmental pathways. Some of the genes that normally help shape the body of a typical seahorse have become altered or inactive. One of the most important developmental systems involved in forming the elongated seahorse profile appears to function differently in the pygmy species. As a result, the familiar horse-like silhouette never fully develops. The snout remains shorter, the body becomes more rounded and irregular, and the final result bears a striking resemblance to the coral polyps surrounding it. Think about what that means. The seahorse did not necessarily become coral-like. by constructing a detailed copy of coral anatomy. Instead, changes in its own developmental program allowed a form to emerge that happened to match the shape of its environment with extraordinary precision. This is where the story becomes philosophically intriguing. The popular image of evolution often suggests that complexity arises through the steady accumulation of new features. We imagine genes acting like engineers, continuously adding new components to an ever more sophisticated machine. But the dwarf seahorse hints at something different. Sometimes a new form emerges, not from adding instructions, but from removing them. Not from construction alone, but from subtraction. Not from writing new lines into the blueprint, but from erasing parts of the existing one. Of course, evolutionary theory can still explain this. Natural selection does not care whether a beneficial trait appears through addition or loss. If a mutation increases survival, it can spread through a population regardless of how it originated. In that sense, the gene-centered framework remains intact. And yet the seahorse raises a deeper question. If development is guided by countless interactions among genes, cells, physical constraints, environmental pressures, and evolutionary history, then perhaps genes alone are not telling the entire story. Perhaps what matters is not merely which instructions exist, but how those instructions interact within a living system and a specific environment to produce form. After all, the final result is extraordinary. A creature so dependent on its coral host that it can barely exist without it. A creature that appears less like an isolated organism and more like a component within a larger ecological structure. The question is no longer simply what genes the seahorse possesses. The question is how a particular form emerges from the interaction of genes, development, environment, and physics itself. And that is where the gene-centered picture begins to give way to something larger, an explanation based not only on information, but on the dynamics through which form discovers itself. At least 4 different kinds of replication come together in the dwarf seahorse, and genes fully describe only one of them. The first is DNA replication. Genes make copies of genes. On this point, Dawkins is absolutely right. The second process is morphogenesis. This is the process through which an entire organism emerges from a single fertilized cell, complete with eyes, skin, a tail, organs, and those distinctive bumps covering its body. Yet there is no miniature seahorse hidden inside the DNA. There is no detailed blueprint specifying where the eye should go, where the fin should form, or how the body should be shaped. There are only chemical instructions, molecular signals triggering other signals in the correct sequence. And somehow, from this cascade of interactions, form emerges on its own, much like a pattern appearing spontaneously in a physical system. The third process is mimicry. The seahorse is not copying itself. It is not even copying its parents. Instead, it is copying a completely different species. Its body conforms to the appearance of the coral with such extraordinary precision that the two seem almost physically connected. No gene explicitly commands the animal to resemble a coral polyp. No instruction says, become coral. The resemblance emerges from the interaction between development, evolution, and environment. It is a convergence of form upon form. And the fourth process is the strangest of all. What appears to be replicated is not the seahorse alone and not the coral alone, but the entire partnership between them. The seahorse cannot survive without its coral host. The coral itself exists within a larger ecological network that shapes the seahorse's existence. What persists through time is not merely an organism, but a relationship. Together they function almost as a single entity, despite belonging to completely different branches of life. Biology can explain the first process. We understand DNA replication in extraordinary detail. We know how genetic information is copied, transmitted, and preserved from generation to generation down to individual molecules and chemical bonds. But genetics alone is not enough to describe morphogenesis, mimicry, or the persistence of these larger biological relationships. To explain those phenomena, we need physics, and not the kind of physics most of us learned in school, not balls rolling down inclined planes or simple equations describing falling objects. We need nonequilibrium statistical physics. It is the science of how a flow of energy moving through matter can spontaneously generate stable patterns. Patterns that persist, adapt, and, in a sense, retain a memory of their past. At first, that may sound abstract, but it is precisely this branch of physics that is beginning to provide mathematical descriptions for processes that we usually think of as exclusively biological. Its goal is ambitious. It seeks to develop a framework that can quantitatively estimate how rapidly evolution should be able to proceed in principle, including the emergence of highly specialized organisms such as bargibanti's dwarf seahorse. And the early results are surprisingly encouraging. When physicists apply these ideas to simplified model systems, their predictions do not conflict with what we actually observe in nature. The equations appear to be pointing in the right direction, which means that bargibanti's dwarf seahorse is far more than an unusual curiosity hiding among coral reefs for divers to photograph. It forces us to confront some of the deepest questions in science. What is life really? What does it mean for something to replicate? Why does matter organize itself into patterns that survive? And why is the universe capable of producing creatures that, in a very real sense, carry models of other creatures within their own bodies? Those questions take us far beyond seahorses, coral reefs, and even biology itself. They lead directly to the boundary where life, information, evolution, and physics begin to merge into a single story. Physicists seek to understand the universe by reducing it to its most fundamental building blocks. We call those building blocks atoms, but far fewer people realize that biology has its own equivalent of the atom. It is the cell. A cell is the smallest unit of matter about which we can say with complete confidence that it is alive. You probably encountered cells for the first time back in school. Perhaps you remember placing a thin slice of onion root under a microscope. Suddenly, a hidden world appeared. Neat rows of tiny compartments stretched across the field of view, arranged in remarkably orderly columns. And maybe, for a brief moment, a strange thought crossed your mind. The onion root pushes through dense soil, anchors the entire plant in the ground, and pulls water upward only because these microscopic structures are constantly growing, dividing, and working together. The world of cells is astonishingly diverse. Most of its inhabitants are far too small to see with the naked eye, yet their sizes vary across an enormous range. Consider parasitic bacteria. Some are so tiny that you could line up 3,000 of their cells end to end and still produce a chain only 1 millimeter long. Now consider a bird's egg. The egg yolk sitting on your breakfast plate is, technically speaking, a single giant cell. Or think about your own body. Some of your nerve cells extend from the base of your spinal cord all the way to the tip of your big toe. A single cell can stretch nearly an entire meter in length. Cell theory is now more than 150 years old. It stands as one of the central foundations of modern biology, yet in school, it is often presented in the most uninspiring way imaginable. We are told that cells are simply the building blocks of life. But the truth is far more fascinating than that. The story began in 1665. The English scientist Robert Hooke built and refined one of the most advanced microscopes of his time. He was not only a brilliant thinker, but also a highly skilled craftsman. One day, Hooke placed a thin slice of cork beneath the lens and peered into a world no human had ever seen before. What appeared before him was a labyrinth of tiny, empty compartments that reminded him of the small rooms occupied by monks in a monastery. He called them cells, meaning little chambers. At the time, Hooke had no idea that he was looking at one of the most important discoveries in the history of science. Soon afterward, the Dutch scientist Antonie van Leeuwenhoek took the next step. Curious about the microscopic world, he scraped plaque from his own teeth and examined it under a microscope. Leeuwenhoek was proud of his personal hygiene, so what he saw came as an unpleasant surprise. His sample was alive. Tiny creatures swarmed in every direction. He affectionately referred to them as little animalcules. They were the first bacteria ever observed and described by a human being. Today, we know that microbes are among the true rulers of this planet. They inhabit almost every environment imaginable, from the upper reaches of the atmosphere to kilometers beneath the Earth's surface. They create fertile soil, recycle dead matter, and provide plants with essential nutrients such as nitrogen. Without microorganisms, complex life on Earth would simply cease to exist. In fact, every human being is far less independent than we tend to imagine. You are not a single organism in the strictest sense. You are a vast and constantly changing ecosystem composed of both human and non-human cells. Trillions of bacteria live on and inside your body, forming intricate communities that influence digestion, immunity, metabolism, and even behavior. By the 19th century, scientists finally began assembling the pieces of the puzzle. The botanist Matthias Schleiden and the zoologist Theodor Schwann combined decades of observations made by researchers across Europe. In 1839, they proposed a revolutionary conclusion. All living organisms are composed of fundamental units that share the same basic nature cells. Later, scientists came to understand something even more remarkable. Cells are not merely passive building blocks. They are living entities in their own right. Some cell lines can survive in laboratories for decades, growing inside ordinary petri dishes. They move, consume nutrients, respond to their surroundings, and continue their existence long after the organism from which they originated is gone. But one major mystery remained. Where do new cells come from? For centuries, many people believed in spontaneous generation, the idea that life could emerge directly from dirt, decay, or non-living matter. It was the German physician Rudolf Virchow who finally brought this debate to an end. He formulated a principle that became one of the foundations of modern biology. Every cell comes from another cell. This simple idea transformed our understanding of life. A single fertilized egg divides again and again, becoming first a cluster of cells and eventually a highly complex human body. Every person who has ever lived began as a single cell. Even now, your body depends on this process. If you cut your finger, cells surrounding the wound begin dividing rapidly, repairing the damage and restoring the tissue. But cell division also has a darker side. When the mechanisms controlling division break down, cells can begin multiplying without restraint. This uncontrolled growth is what we call cancer. As one medical text put it, growth, regeneration, degeneration, and malignant tumors are all connected to changes in the properties of cells in health and disease, in youth and old age. The roots of many illnesses can ultimately be traced to disruptions in cellular function, and understanding those disruptions is the foundation of developing effective treatments. In the 1970s, a new generation of geneticists began investigating cell division using one of the simplest organisms imaginable, yeast. At first glance, yeast seems almost trivial. Most people associate it with bread or beer. Yet, yeast is a fungus, and its cells share many important features with our own. It grows rapidly, costs very little to maintain, and is remarkably easy to study in the laboratory. Those humble yeast cells transformed modern biology by observing them under controlled conditions. Scientists uncovered fundamental mechanisms governing cell growth and division. Much of what we know today about the cell cycle and much of the knowledge that underpins modern cancer research can be traced back to experiments performed on these simple organisms growing quietly inside petri dishes. A cell is an incredibly complex structure. Inside it, entire microscopic industries are at work. Tiny power stations generate energy. Molecular factories manufacture proteins. Transport systems move materials from one location to another. Biologists call these internal structures organelles. At the center of the cell sits the nucleus. This is the primary command center. Inside it are chromosomes containing the genetic instructions that govern the cell's existence. Throughout the cell float mitochondria, the cell's miniature power plants. They produce virtually all the energy required for life. To make this possible, the cell relies on an elaborate transport network and logistics system that constantly builds, breaks down, packages, and delivers the materials needed to keep the entire system running. The presence of a nucleus divides all life on Earth into two vast biological empires. Organisms whose cells contain a nucleus are known as eukaryotes. This group includes plants, fungi, animals, and humans. Organisms whose cells lack a nucleus are called prokaryotes. These include bacteria and archaea, some of the oldest and most resilient forms of life on the planet. But perhaps the most important structure of all is the cell membrane. This membrane is only a few molecules thick, yet this unimaginably thin barrier separates the inner universe of the cell from the outer chaos surrounding it. And that barrier carries profound physical significance. According to the laws of thermodynamics, the universe naturally tends toward disorder. Entropy increases, structure breaks down, order dissolves. Left alone, everything drifts toward randomness. The cell does the opposite. Within its delicate, lipid walls, it continuously creates and maintains an extraordinary degree of order. Molecules are arranged in precise locations. Reactions occur at exactly the right moments. Resources are allocated where they are needed. Damage is repaired. Information is stored and transmitted. To preserve this internal order, the cell constantly exports disorder into the environment around it. In other words, life survives by creating islands of order in a universe that prefers chaos. But cells do not merely exist as passive structures. They sense even the slightest changes in their surroundings and respond to them. They detect nutrients, toxins, temperature shifts, chemical signals, and threats. They alter their behavior, reorganize their internal machinery, and adapt to changing conditions. From the outside, this behavior can seem almost purposeful. A cell acts as though it possesses an unwavering obligation: survive at all costs, reproduce whenever possible, and pass its existence into the future. To understand where this apparent purpose comes from, we must look inside the nucleus itself, because that is where the instructions for this entire mechanism are stored. Think about your own family. Chances are you are all very different people. Some may work in finance, others in law, others with their hands. Your personalities may differ, your interests may clash, and your lives may have taken completely different paths. And yet, when the entire family gathers in one place, something immediately becomes obvious. You belong to the same lineage. The shape of the eyes, the curve of the smile, the structure of the face, even certain expressions and gestures seem to echo across generations. The resemblance between parents and children has fascinated human beings for thousands of years. Aristotle wondered about it as well. Admittedly, his explanation sounds rather strange today. He believed that during reproduction, the father supplied the active principle, the seed, while the mother merely provided the environment in which it could grow, much like soil nourishing a plant. Other thinkers proposed a different idea, the theory of blended inheritance. According to this view, the traits of the parents simply mix together like paints, producing an unpredictable combination in the child. Everything changed when science began uncovering the mechanisms of heredity and eventually discovered the existence of genes, the fundamental instructions that guide the construction of cells and the operation of living organisms. The first person to truly uncover the logic behind inheritance was an Augustinian monk and schoolteacher named Gregor Mendel. Curiously, he did not study humans at all. Instead, he conducted experiments on pea plants. Before Mendel, plant breeders had already noticed strange patterns. For example, crossing a purple flower with a white one could sometimes produce pink offspring. Observations like these seem to support the idea that parental traits simply blended together. But nobody could explain the deeper logic hidden beneath those results. Mendel showed that blending was only part of the story and often not the correct story at all. For many traits, inheritance followed an entirely different set of rules. What made Mendel unusual was his background. He had studied mathematics and physics, working under the influence of the physicist Christian Doppler, yes, the same Doppler whose name survives today in the Doppler effect. Unlike most naturalists of his era, Mendel approached biology with the mindset of a physicist. He believed that if nature followed rules, then those rules should reveal themselves through numbers. To uncover those rules, he realized he would need enormous amounts of data. His experiments involved tens of thousands of pea plants. No one before him had attempted breeding studies on such a scale. Mendel selected traits that could be measured clearly and objectively: seed, shape, flower, color, pod, structure, and several others. Year after year, he carefully crossed plants, counted offspring, recorded outcomes, and filled notebooks with observations. Then, something extraordinary happened. Patterns emerged. Not vague patterns, not impressions, mathematics. Again and again, Mendel found the same numerical ratios appearing in his results. Eventually, he concluded that pollen and ovules must carry discrete hereditary units, hidden factors that pass traits from one generation to the next. Today, we call those factors genes. What makes the story even more remarkable is that at roughly the same time, Charles Darwin was studying breeding experiments involving snapdragons and observing similar inheritance patterns. The evidence was right in front of him. But Darwin was not thinking like a mathematician. Mendel was. And because he searched for a formula where others saw only observations, he became the first person to glimpse the hidden code of heredity. Mendel did exactly that. He found the mathematics hidden inside inheritance. But there was a problem. Almost nobody paid attention. His work was published, discussed briefly, and then largely forgotten. For decades, the scientific world moved on as if his discoveries had never happened. Then, in 1900, something remarkable occurred. 3 scientists working independently, Hugo de Vries, Carl Correns, and Erich von Tschermak, arrived at conclusions strikingly similar to Mendel's. As they reviewed the scientific literature, they made an embarrassing discovery. A monk working in a monastery garden had already solved the problem 34 years earlier. Overnight, Mendel's work was resurrected. And with it, an entirely new science was born: genetics. The central law of inheritance turned out to be surprisingly simple. Genes exist in pairs. One copy comes from the mother, the other comes from the father. At the moment of conception, those copies are brought together and become part of a new organism. By the end of the 19th century, advances in microscopy allowed biologists to observe cell division in unprecedented detail. For the first time, they could actually watch inheritance unfold before their eyes. One of these researchers was the German physician Walther Flemming. While studying dividing cells, he noticed thin, thread-like structures inside the nucleus. Just before division, these threads condensed, became clearly visible, and then split into identical halves. One half moved into one daughter cell, the other half moved into the second. Today, we call those structures chromosomes. Chromosomes are the physical carriers of genetic information. They are the structures that contain the genes Mendel had predicted decades earlier through mathematics alone. Soon afterward, the Belgian scientist Eduard van Beneden chose an unusual organism for study: roundworms. It was a brilliant decision because these worms possess only 4 chromosomes, making them far easier to analyze than more complex organisms. Van Beneden observed that the first cell of the embryo contained exactly 4 chromosomes. 2 had come from the mother, 2 had come from the father. Suddenly, Mendel's abstract laws acquired a physical form. This is one of the great lessons of science. Simple systems often reveal the deepest truths. The universal laws governing nature are usually easiest to see when unnecessary complexity has been stripped away. That is why biologists frequently study yeast, worms, fruit flies, and bacteria before attempting to understand human beings. Simple models make fundamental principles visible. The number of chromosomes varies enormously. Across life, every P cell contains 14 chromosomes. Human cells usually contain 46, though in some rare conditions there may be 47. Certain species of blue butterflies possess more than 400. Almost every cell in your body contains chromosomes. The major exception is the mature red blood cell, which eventually loses its nucleus. Most of your other cells, however, carry the complete genetic instructions required to build and maintain your body. And every time a cell divides, those instructions must be copied with astonishing accuracy. But this raised an even deeper question. What exactly are genes made of? For decades, nobody knew the answer. Then, in 1944, a breakthrough arrived in New York. In the 1940s, microbiologist Oswald Avery and his colleagues were studying the bacteria responsible for pneumonia. Researchers had already discovered a strange phenomenon. If harmless bacteria were mixed with the remains of dead, disease-causing bacteria, the harmless microbes could suddenly become dangerous themselves. Even more remarkably, this new trait was passed on to their descendants. Something was being transferred from the dead bacteria to the living ones. Avery suspected that some chemical substance carried the instructions responsible for this transformation. Whatever this substance was, it permanently altered the hereditary properties of the receiving bacteria. After years of painstaking work, Avery and his team isolated the mysterious component. It was deoxyribonucleic acid, DNA. The result shocked the biological community. At the time, DNA was considered a relatively simple and uninteresting molecule. Most scientists believed that genes had to be made of proteins because proteins were far more complex and seemed much better suited to storing biological information. But the evidence pointed elsewhere. Avery demonstrated that hereditary information was carried by DNA itself. Genes were not proteins. Genes were DNA. This discovery transformed biology. Scientists now understood that every chromosome consists of a single, enormous DNA molecule. Along its length, genes are arranged one after another, like cars connected along a train. Consider human chromosome 2. It contains roughly 1,300 genes. If the DNA molecule inside that chromosome was stretched into a straight line, it would measure approximately 8 centimeters in length. Nature somehow manages to coil, fold, twist, and package that strand tightly enough to fit inside a microscopic cell nucleus. And that is just one chromosome. A single human cell contains roughly 2 meters of DNA. Pause for a moment and think about that. Inside every microscopic cell of your body lies a thread of genetic material, longer than you are tall. Now multiply that by the trillions of cells that make up your body. If all the DNA in your body were connected end to end, the resulting strand would stretch for approximately 60 billion kilometers. That distance is enough to travel from Earth to the Sun and back more than 20 times. Oswald Avery himself remained a remarkably modest man. He did not celebrate his achievement loudly, and many scientists continued criticizing his conclusions for years. But history ultimately proved him correct. Genes turned out not to be some mysterious life force. They were physical structures, stable arrangements of atoms, molecules that obey the same laws of chemistry and physics as everything else in the universe. Then, in 1953, biology entered an entirely new era. Scientists finally uncovered the structure of DNA itself. Most major discoveries develop slowly. Researchers spend years sorting through confusing evidence, false leads, and competing ideas before a clear picture emerges. But in this case, events moved with astonishing speed. At King's College London, Rosalind Franklin and her graduate student Raymond Gosling were producing the most detailed X-ray diffraction images of DNA ever obtained. Among them was the image that would later become famous around the world, Photo 51. At the same institution, Maurice Wilkins was conducting his own DNA research. Meanwhile, in Cambridge, 2 young scientists, Francis Crick and James Watson, were attempting to solve the puzzle from another angle. Rather than studying DNA directly, they were building physical models of the molecule, trying to uncover the hidden structure that lay behind life itself. In early 1953, a crucial moment occurred in one of the greatest scientific races of the 20th century. Maurice Wilkins showed Watson the now famous Photo 51, without Rosalind Franklin's knowledge. This image, together with Franklin's unpublished measurements and calculations, provided Watson and Crick with several of the critical parameters they needed to complete their model of DNA. A few months later, in April 1953, the journal Nature published 3 papers side by side. The first presented Watson and Crick's double helix model. The second contained the X-ray diffraction data collected by Wilkins and his collaborators. The third featured the independent work of Rosalind Franklin and Raymond Gosling, whose experimental results strongly supported the structure proposed by Watson and Crick. The discovery transformed biology almost overnight. Nearly a decade later, in 1962, the Nobel Prize in Physiology or Medicine was awarded to Watson, Crick, and Wilkins for their discoveries concerning the molecular structure of DNA and its significance for the transfer of information in living systems. Rosalind Franklin was absent. She had died 4 years earlier in 1958 at the age of only 37. Most historians believe that years of exposure to X-ray radiation during her research likely contributed to the ovarian cancer that took her life. Because Nobel Prizes are not awarded posthumously, Franklin never received the recognition that many scientists believe she deserved. But the true beauty of the double helix lies not merely in its elegance. The structure itself explains 2 of the most fundamental requirements of life. First, DNA must be capable of storing enough information to build, maintain, and reproduce an entire living organism. Second, it must be capable of copying that information with extraordinary accuracy. The double helix accomplishes both. Imagine DNA as a twisted rope ladder. The sides of the ladder are formed by repeating molecular chains, while each rung consists of a pair of chemical building blocks known as nucleotides. There are only 4 of these nucleotides: A for adenine, T for thymine, G for guanine, and C for cytosine. That's it. Every living organism on Earth is written using just 4 molecular letters. The sequence of those letters forms the genetic code. A gene is simply a particular stretch of that code containing instructions the cell can use. One gene may contribute to the color of a flower's petals. Another may help determine how a bacterium infects its host. Another may influence the development of an eye, a muscle, or a nerve cell. The cell reads these instructions and acts upon them. And DNA's method of replication emerges directly from its geometry. A can pair only with T. G can pair only with C. Those rules are absolute. If the 2 strands of the double helix are separated, each strand automatically becomes a template for rebuilding its missing partner. The information needed to create a copy is already embedded within the structure itself. In this way, DNA solves one of the deepest problems of life: how information can persist across generations. But genes do not build cells directly. Genes contain instructions for constructing proteins, and proteins perform most of the work inside the cell. They form structural components, act as molecular machines, regulate chemical reactions, transport materials, and allow cells to communicate with one another. Much of what a living cell actually does depends on proteins, and this created a new mystery. DNA stores information using a 4-letter alphabet. Proteins are built from 20 completely different molecular building blocks known as amino acids. How does a cell translate information from one language into another? For years, this question remained one of biology's greatest cryptographic puzzles. The code was finally cracked during the 1960s, thanks to the work of several researchers. One of the key figures in that breakthrough was Sidney Brenner. Of all the biologists working on the genetic code, Sidney Brenner was perhaps the boldest and certainly one of the most brilliant. Together with his colleagues, he helped demonstrate that DNA is read in 3-letter units. The genetic alphabet is not interpreted 1 letter at a time, but in groups of 3, known as codons. Take the human beta-globin gene as an example. It contains 441 genetic letters. Those letters are organized into 147 3-letter words. The cell reads each word and translates it into a specific amino acid. One after another, 147 amino acids are assembled into a protein chain. The result is beta-globin, one of the key proteins that allows our blood to carry oxygen and gives it its characteristic red color. When scientists finally cracked the genetic code, many believed the greatest mysteries of biology had been solved. Francis Crick himself became so confident in the success of molecular biology that he largely left genetics behind and turned his attention to one of the deepest puzzles of all: human consciousness. But that confidence proved premature. Looking back from the perspective of the 21st century, we now understand that deciphering DNA was not the end of the story. It was the beginning. Over the following decades, the very concept of a gene underwent a profound transformation. Genes ceased to be abstract theoretical entities and became tangible sequences of chemical letters that could be identified, mapped, manipulated, and studied directly. A gene was no longer just an idea, it was a physical object. As a result, geneticists gradually became molecular engineers. They learned how to locate genes within chromosomes. They learned how to isolate them, copy them, modify them, and transfer them between entirely different organisms. In the late 1970s, researchers achieved something that would have seemed almost miraculous only a few years earlier. They extracted the human insulin gene and inserted it into the DNA of an ordinary bacterium, Escherichia coli. The bacterium became a living factory. Instead of producing only its own proteins, it began manufacturing an exact copy of a human one. The implications were enormous. Before this breakthrough, insulin for diabetic patients had to be extracted from the pancreases of pigs and cattle. Now, bacteria could produce pure human insulin in vast quantities. Since then, genetically engineered insulin has saved millions of lives around the world. Then came another revolution. The British biochemist Frederick Sanger developed a method for reading DNA letter by letter. For the first time, scientists could determine the exact sequence of nucleotides within a gene. Sanger was known as an unusually modest and thoughtful scientist, but the significance of his invention was impossible to ignore. The achievement earned him a second Nobel Prize, making him one of the very few people in history to receive the award twice. The pace of discovery accelerated dramatically. By 2003, humanity completed one of the most ambitious scientific projects ever attempted: the Human Genome Project. For the first time, we possessed a complete draft of our own genetic blueprint, roughly 3 billion letters of DNA. Within that vast sequence, researchers identified around 20,000 human genes. It was an extraordinary accomplishment. Yet, almost immediately, a new problem emerged. A list of genes is not the same thing as understanding them. Knowing the sequence of letters is one thing. Understanding precisely what those letters do, how they interact, and how they produce a living organism is something else entirely. The bridge between the genome and the living cell is the process of cell division. Every time a cell divides, its chromosomes must first be copied and then distributed equally between the two daughter cells. The process must occur with astonishing precision. Even small mistakes can have severe consequences. In many cases, cells respond to serious errors by triggering apoptosis, programmed cell death, a built-in mechanism that prevents damaged cells from spreading. Coordinating all of this is one of the most sophisticated control systems in biology: the cell cycle. The cell cycle consists of 4 major phases. During the S phase, the cell duplicates its DNA, creating an exact copy of every chromosome. During mitosis, those duplicated chromosomes are separated and distributed between the 2 new cells. Between these stages lie 2 growth phases, during which the cell increases in size, monitors its internal condition, repairs potential problems, and ensures that everything is ready before moving on to the next step. Only when every checkpoint has been passed does the cycle continue. All of this is extraordinarily complex chemistry. But life begins at the moment when hundreds, thousands, and ultimately millions of individual chemical reactions become organized into a functioning system. A living cell is not merely a collection of molecules. It is a coordinated process. In a very real sense, the cell cycle is what brings otherwise lifeless molecules into something that behaves as though it has purpose. Remarkably, the cell cycle is controlled by genes themselves. To identify the genes responsible, scientists turned once again to one of biology's favorite model organisms, ordinary yeast, the same organism used to brew beer and bake bread. Yeast may seem simple, but the fundamental logic of its cell cycle is astonishingly similar to our own. The same core mechanisms that govern division in yeast also operate in human cells. Researchers searching for these mechanisms are particularly interested in mutant cells carrying random changes in their DNA. The word mutant often conjures images of monsters, deformities, or science fiction creatures. In genetics, however, the term means something much simpler. A mutant is merely an organism carrying a variation in its genetic code. In fact, many of the differences we observe between people are the result of mutations. Eye color, for example, is ultimately determined by genetic variations that accumulated over generations. Most mutations are completely harmless. They are simply alternative versions of existing genes. Take the ability to drink milk as an adult. For most of human history, adults lost the ability to digest lactose after childhood. The enzyme responsible for processing milk sugar was switched off after weaning. Then, at some point in the distant past, a mutation altered regulatory instructions controlling that enzyme. As a result, it remained active throughout life. The descendants of those early mutants gained a new ability. Today, hundreds of millions of people can drink milk without difficulty because they inherited that genetic change. Another famous example comes from the Italian village of Limone sul Garda. There, researchers discovered a rare mutation affecting a protein involved in fat transport through the bloodstream. The altered protein is remarkably effective at clearing fats from blood vessels. People who carry this mutation show unusually low rates of atherosclerosis, even when consuming diets rich in fat. The effect is so powerful that scientists have spent years trying to turn the biological mechanism behind it into a medical treatment. There is also a mutation that provides resistance to HIV. Some individuals, particularly in parts of Northern Europe, carry a defective version of a gene called CCR5. Under normal circumstances, CCR5 produces a receptor on the surface of immune cells. HIV uses this receptor as an entry point, a kind of molecular door handle that allows the virus to enter the cell. In people carrying the mutation, that door handle is missing or altered. The virus arrives, but the door remains closed. As a result, these individuals possess a remarkable degree of resistance to HIV infection. Fortunately, mutations are relatively rare. DNA replication is one of the most accurate processes known in nature. Every time a cell divides, roughly 3 billion genetic letters must be copied. Yet the error rate is astonishingly low, only about 3 mistakes per billion letters copied during a typical cell division. Still, mistakes do occur. Ultraviolet radiation, chemical damage, random copying errors, or environmental stress can alter the sequence of DNA. Sometimes the consequences are minor, sometimes they are profound. In certain cases, changing a single letter in a gene is enough to transform an entire protein. One famous example is sickle cell anemia. A mutation affecting just one letter in the beta-globin gene alters the structure of hemoglobin, the protein responsible for carrying oxygen through the blood. That tiny change produces a serious and potentially life-threatening disease. This illustrates a fundamental truth about life. Genes survive across generations because they are copied with extraordinary precision. The entire history of life depends on preserving information accurately enough to withstand the passage of time. And nature has become exceptionally good at that task. The genome of almost any person on Earth is more than 99.9% identical to yours. In genetic terms, human beings are astonishingly similar. We remain remarkably close to the people who sat around campfires tens of thousands of years ago, telling stories beneath the night sky. The overwhelming majority of our genetic code has remained unchanged across vast stretches of time. Yet the remaining fraction makes all the difference. The human genome contains roughly 3 billion genetic letters. At first glance, a difference of just 0.1% sounds insignificant. But 0.1% of 3 billion is still about 3 million letters. Hidden within those 3 million variations are countless details that make you who you are. The color of your eyes, the shape of your face, your height, your metabolism, your susceptibility to disease, aspects of your personality, and even the sound of your voice. One person differs from another by only a tiny fraction of their DNA. And yet that tiny fraction is enough to produce 8 billion unique individuals. The remaining 99.9% unites us. The remaining 0.1% defines us. But genetics is not always kind. Sometimes a single altered letter can completely change the course of a person's life. In southern Ecuador, for example, there is a population of roughly 100 individuals who rarely grow taller than about 1.2 meters. The cause is a mutation affecting the receptor for growth hormone. Their bodies produce the hormone normally, but their cells cannot properly receive the signal telling them to grow. At first glance, this appears to be a severe disadvantage. Yet the mutation comes with an extraordinary side effect. These individuals almost never develop cancer. They also show unusually low rates of diabetes. As a result, scientists around the world have studied them intensely, hoping to uncover clues that could one day help us combat aging and age-related disease. Other mutations are far less dramatic. Some are simply beautiful. Consider blue eyes. Around 10,000 years ago, somewhere near the Black Sea region, a mutation disrupted the production of brown pigment in the iris. The result was a new eye color that had never existed before. Every blue-eyed person alive today is believed to share a common ancestor carrying that original mutation. The same is true of red hair. It too emerged from a small alteration in a gene involved in pigment production. A simple genetic typo created one of the most distinctive features in human appearance. Then there are mutations that seem almost superhuman. Scientists have identified rare individuals carrying mutations that dramatically increase bone density. Their skeletons become unusually strong and resistant to fracture. In one famous case, a man survived a serious car accident without suffering a single broken bone. Researchers later discovered that his extraordinary resilience was linked to a mutation affecting bone growth. Nature is constantly shuffling the genetic deck. Most of the time, the changes are subtle. Occasionally, something extraordinary appears. Every person born today carries a collection of new mutations that did not exist in either parent. These changes arise naturally as DNA is copied from one generation to the next. Most have little effect. Some disappear. A few spread through populations. Without them, evolution would stop. Life would become trapped in its current form. Mutations are the mechanism through which nature experiments. They provide the raw material from which adaptation, innovation, and entirely new forms of life emerge. Genes, therefore, exist in a delicate balance. They must be stable enough to preserve billions of years of accumulated biological knowledge. At the same time, they must remain flexible enough to adapt to a changing world. Too much stability, and life cannot evolve. Too much change, and information is lost. It is this extraordinary balance between permanence and variation that has produced the breathtaking diversity of life on Earth. Now we are ready to consider a fundamental question. How does non-living matter become alive? How does all this biological diversity emerge? To understand how dead matter becomes more complex and acquires behavior, we need to look at genes in a new way. So far, we have described genes as instructions. That is a useful metaphor, but it conceals something deeper. Physicist David Deutsch suggests a different perspective. A gene is a form of knowledge. Not knowledge in the sense of conscious understanding, but in the most basic, physical sense. A gene is a record of how the environment was structured in which its carriers survived and reproduced. Consider a simple example. The rings inside a tree trunk record past droughts, wet seasons, and favorable years. DNA is a similar kind of record, but on the molecular level, and concerning far more subtle things: how to transport oxygen, what temperatures can be tolerated, and how to fold a protein so that it functions correctly in a particular environment. Over billions of years, natural selection has transformed DNA into a densely packed chronicle of matter's interaction with the world. Viewed this way, we stop thinking of genes as the cause of life and begin to see them as its result. Physicists have proposed a compelling hypothesis. According to this idea, life is matter through which energy flows. And those configurations of matter that allow energy to pass through them most effectively are more likely to persist and reproduce their structure. Those are the configurations we call genes. Genes are simply matter in which information has been encoded about how to allow energy from the environment to flow through without the structure falling apart. And this raises a question that biology cannot answer using only its own tools. How can ordinary matter, in principle, acquire information about its environment? This is no longer a question about DNA. It is a question for physics, more specifically for the physics of open systems through which energy flows. And it is precisely this branch of physics that has begun to provide the first outlines of an answer in recent years. That answer is connected to the work of physicist Jeremy England, who is sometimes referred to as the new Darwin. He proposed a concept known as dissipative adaptation. The nickname emerged after Pulitzer Prize-winning science historian Edward Larson remarked that if England could prove his hypothesis, he might become the next Darwin. England himself rejects that characterization and considers it an exaggeration. He describes his theory not as a replacement for Darwinian evolution, but as a possible extension of it. All living things are made of atoms, and atoms obey strict physical laws. From this, it is tempting to conclude that life is simply a byproduct of the laws of nature. This is, in many ways, the physicist's perspective, and it has been extraordinarily successful. Using this approach, scientists have learned to examine DNA and proteins in remarkable detail. They have deciphered the genetic code, uncovered the molecular machinery of the cell, and even learned how to reprogram living organisms. It can seem as though we are only a few steps away from explaining biology entirely in terms of physics. But if we approach the mystery of life only in this way, we will fail. And there are two important reasons why. The first reason is what we might call the illusion of reductionism. It is the belief that the universe works like a mechanical clock. If we understand every individual gear and spring, then surely we should be able to predict the behavior of the entire machine. For centuries, science achieved remarkable successes using exactly this strategy. Newton explained gravity. Einstein transformed our understanding of space, time, and light. Schrödinger described the behavior of electrons through mathematics. These were extraordinary triumphs, but they share a limitation. They work best for systems that are simple and isolated. A planet orbiting a star, an atom suspended in a vacuum, a small number of interacting components. The moment additional complexity enters the picture, the situation changes dramatically. Add a third body to a gravitational system, and you encounter the famous three-body problem. Suddenly, prediction becomes enormously difficult. Newton's laws have not stopped working, but applying them becomes almost impossible in practice. The same is true elsewhere. Fundamental physical laws cannot easily predict a stock market crash, the exact motion of a turbulent river, or the behavior of an ecosystem containing thousands of interacting species. As complexity increases, the explanatory power of simple reduction begins to weaken. The second mistake lies in the language we use. Many people assume that a perfect physical theory will automatically tell us what life is, just as physics explains why water freezes or why planets orbit stars. But that expectation is misleading. Physics cannot define life for us, because life is not a physical quantity. It is a concept created by human beings. Before physics can explain life, we must first decide what we mean by the word itself. We must determine where we choose to draw the boundary between the living and the non-living. Only then can physicists attempt to describe that boundary using equations and physical principles. And one of the simplest places to see the limits of reductionism is not in a laboratory. It is in an ordinary kitchen. Imagine a pot of water sitting on a stove. We know that when heated, water eventually boils. A strict reductionist might say, water is made of molecules. Let's simply write down the Schrödinger equation for every atom in the pot and calculate the exact moment when the liquid turns into steam. But that plan is doomed from the start. To calculate the behavior of such a vast number of particles with complete quantum precision, we would need a computer of unimaginable size, one that would not even fit within our galaxy. The direct path from the microscopic world to the macroscopic one is effectively closed to us. Fortunately, we do not need perfect precision. Nature provides something far more useful. It is called a phase transition. And phase transitions beautifully demonstrate why tiny changes in microscopic details can produce dramatic changes on a large scale. Take water. At 98 degrees Celsius, it is an extremely hot liquid. At 102 degrees Celsius, it has become a gas. If we measured the average speed of the molecules, we would find that their velocity increased by less than 1%. Less than 1%. So how can such a tiny change transform the entire substance? The answer lies in collective behavior. Water molecules attract one another, but when a molecule moves just slightly faster than usual, it can break free from those attractive forces. And what matters is not the behavior of one molecule, but the behavior of billions upon billions of them acting together. This is one of the central ideas of modern physics. New properties emerge through the interaction of many particles. And those properties cannot be predicted simply by studying a single atom in isolation. Inside a liquid, molecules exist in extreme crowding. They constantly collide, jostle, and move in random directions. Yet despite all that motion, they remain together because attractive forces continuously pull them back toward one another. In a gas, the situation is completely different. There, particles move at tremendous speeds. They fly past one another so rapidly that they rarely remain close enough to form lasting associations. As a result, the same amount of matter suddenly occupies an enormous volume. The transition from liquid to gas can be thought of as a kind of mass migration. Imagine, for a moment, that every molecule has a fixed speed. Then, the question of escape becomes a matter of overcoming a kind of potential well. The more neighboring molecules surround a particle, the more strongly they hold it in place. It is as though the molecule sits at the bottom of a deep valley. But if one particle manages to break free, the grip weakens. A second escapes. Then a third. A chain reaction begins. At the boiling point, water exists on a knife's edge. The slightest microscopic shift in the forces of attraction can trigger a full-scale exodus. Molecules rush collectively into the vapor state. Because of this extraordinary sensitivity to detail, computer simulations of such processes become incredibly challenging. The smallest error in calculating intermolecular forces can cause even the most powerful supercomputer to confidently predict that steam is liquid or liquid is steam. Our world is filled with collective effects of this kind. Phase transitions are merely the simplest examples. Whenever vast numbers of particles interact, their collective behavior can change dramatically in response to tiny alterations in the underlying rules. This is precisely where strict reductionism begins to fail. You cannot understand the behavior of an ocean by studying a single drop of water. As physicist Philip Anderson famously put it, more is different. Even a perfect theory of atoms cannot, by itself, explain the viscosity of honey or the intricate geometry of a snowflake. Those properties do not exist within individual atoms. They emerge from immense collections of them acting together. Fortunately, physicists do not need to track every one of the trillions upon trillions of molecules involved. They have found a shortcut. When a liquid turns into a gas, its volume changes dramatically and it absorbs heat. By measuring these quantities, we can predict how the boiling point will shift under different temperatures and pressures. To do this, physicists use a powerful mathematical relationship known as the Clausius-Clapeyron equation. And here is the remarkable part. To use this equation, you do not need to know anything about quantum mechanics. You do not need to know the exact structure of the atoms involved. The microscopic details become irrelevant. Consider another example. Suppose you fire a bullet into a large tank of water. We can calculate with extraordinary precision how much the water will heat up. All we need to know is the bullet's mass, its velocity, and the amount of water in the tank. A first-year physics student can solve this problem without knowing the bullet's shape, the arrangement of water molecules, or the microscopic details of the collision. Why? Because the law of conservation of energy does all the work. The bullet's kinetic energy is transformed into thermal energy. We do not need to know precisely how billions of water molecules collide and rub against one another. Only the overall energy balance matters. This insight gave birth to thermodynamics, the science of making precise predictions in systems whose microscopic behavior is overwhelmingly complex. And perhaps the most surprising thing is that the same mathematical framework describes phenomena that seem completely unrelated. The boiling of water, the magnetization of iron, the melting of ice, the self-assembly of molecules into a cell membrane. On the mathematical level, these are all variations of the same underlying process. Physicists refer to this phenomenon as universality. During the 20th century, its rigorous description became one of the great achievements of theoretical physics and led to multiple Nobel Prizes. But if the same principles can describe boiling water, magnets, crystals, and membranes, what about life? We look at a living organism with much the same sense of wonder that a child feels while watching an ice cube melt. A living creature is made of the same dead atoms as everything else around us, and yet it differs profoundly from non-living matter. It is natural to ask whether life itself might simply be another kind of phase transition. In the middle of the 20th century, this question fascinated some of the founders of quantum mechanics. Niels Bohr felt that there was something fundamentally special about life, something that could not be fully reduced to the sum of its atoms, although he could never prove it. Erwin Schrödinger approached the problem more directly in his famous book, What is Life? In that book, Schrödinger made two remarkably insightful predictions. First, he proposed the existence of a special hereditary molecule, which he described as an aperiodic crystal. Today, we know that molecule as DNA. Second, he argued that living systems sustain themselves by feeding on what he called negative entropy. Yet even Schrödinger left room for mystery. On one hand, he was convinced that living cells obey the laws of physics. On the other, he clearly recognized that life behaves very differently from any other form of matter known to science. And so, the question remains, can physics fully describe life? Over the past 50 years, molecular biology has established one fact beyond any reasonable doubt. Every capability of a living organism ultimately depends on the physical properties of its material components. There is no hidden life force beyond atoms, molecules, and their interactions. But that does not mean life can be reduced to simple physics. If anything, the opposite seems true. Everywhere we look in biology, we encounter emergent properties, features that arise from the interaction of many components and cannot be understood by examining those components individually. Consider blood. Blood is a fluid that circulates through the body, transporting oxygen to every tissue. At the molecular level, we understand quite well how hemoglobin binds oxygen and later releases it. This behavior can be explained directly from the atomic structure of the protein. But now try to calculate the exact viscosity of blood, starting from the fundamental laws of physics. You cannot. There are simply too many interacting components. Cells collide with one another. Molecules interact continuously. Countless microscopic factors influence the result. Even tiny changes can alter the behavior of the system. In practice, no computer can predict blood viscosity as accurately as a straightforward experiment. Life exists in a world of extraordinary fine-tuning. Some of its properties can be derived directly from physics. We can calculate, for example, how a hemoglobin molecule binds oxygen. But other properties, such as the viscosity of blood or the precise shape of a chromosome during cell division, cannot be predicted from first principles. They emerge from the interaction of vast numbers of components. And in such systems, even the smallest deviation can have enormous consequences. Change the viscosity of blood by 20% and a person may die. Change a single letter in DNA and you can end up with sickle cell anemia. This is where the divide between physics and biology emerges. And it is not a temporary gap that will someday be closed by a more powerful computer. It is a difference in language itself. The two sciences literally describe the world using different vocabularies. Physics measures. Its language is made of numbers. Distance in meters, mass in kilograms, temperature in degrees. Every physical phenomenon is expressed through measurable quantities and the relationships between them. When Newton formulated his second law of motion, that is exactly what he did. He connected three independently measurable quantities with a single equation. And it is this precision that gives physics its extraordinary power. Biology is built differently. Of course, modern biology uses mathematics, statistics, and computation. But at its core, biology is not fundamentally quantitative. It begins by accepting the distinction between living and non-living things, and then seeks to understand the properties and behavior of living systems. A biologist does not need an equation to recognize that a bee pollinates a flower, that a tree grows towards sunlight, or that a mother feeds her offspring. None of these statements contain numbers. Yet all of them describe something essential about life. Their foundation is observational rather than mathematical. Physics does not work that way. Remove the numbers from a physical law, and very little remains. We do not learn the meaning of life from a textbook because we are already participants in it. We encounter living things every day in the neighbor's dog, in the mold growing on a forgotten piece of bread, and in our own bodies. Science refines this understanding, but it does not create it from scratch. The concept of life did not originate in physics, and physics does not get to define its meaning. This is why the unease felt by Bohr and Schrödinger could never be resolved by physics alone. Their question ultimately belonged to philosophy as much as science. One of their contemporaries, the philosopher Ludwig Wittgenstein, attempted to address exactly this problem. He famously wrote, "The limits of my language mean the limits of my world." The same event can be described in entirely different ways depending on the language we choose to use. Imagine a drop of rain being absorbed by the root of a tree. A physicist sees capillary action, osmotic pressure, molecular forces, and gradients of energy. A biologist, meanwhile, would simply say that the tree is drinking. Both descriptions are correct. They are simply different languages describing the same reality. This is why a theory built entirely from physical concepts will never identify the exact moment when life begins. The word life was not invented by physicists, and it is not for physicists alone to determine what it means. There is nothing wrong with this division of languages. Physical measurements do not replace biological concepts. They make them more precise. Saying that a person is alive and saying that their temperature is 36.6 degrees Celsius, their pulse is 70 beats per minute, and their blood pressure is 120/80 are different kinds of statements. Yet there is a clear bridge between them. A biologist identifies a state, a physicist measures it. Neither approach works particularly well without the other. Much of modern biophysics operates exactly this way. It studies living systems through their material properties. Consider hemoglobin, the protein that carries oxygen through our blood. It is an enormous molecule composed of thousands of atoms arranged into 4 polypeptide chains. Its specific three-dimensional structure allows it to bind oxygen molecules and transport them throughout the body. Without that structure, life as we know it would be impossible. But notice something important. We call hemoglobin useful because we already know what it is for. We know that it transports oxygen. Now, imagine an alien physicist encountering this molecule for the first time with no knowledge of blood, lungs, or respiration. They would see an intricate and highly ordered structure, but they would not automatically understand its significance. To recognize that a molecule functions as a tool, you first have to know the problem it solves. And a problem is not a physical object. Biophysics usually begins with a biological question and then studies the physical mechanisms that provide the answer. But that leaves a deeper mystery untouched. How did purpose arise in the first place? How does non-living matter acquire function without an external designer? The answer appears to lie in structure. If atoms are assembled completely at random, useful tools do not emerge. Highly specific and functional arrangements of matter are extraordinarily rare. We intuitively recognize this rarity whenever we observe the complexity of an organ, a protein, or even a single cell. To understand the transition from the non-living to the living, physics must explain how these rare and highly organized structures emerge from ordinary matter. Life may not have a single, universal, physical definition, but it does seem to possess a set of recognizable physical characteristics. Living systems do 3 unusual things. They copy themselves, they extract energy from their environment, and they preserve information about that environment, embedding into their structure traces of what has worked in the past. Each of these abilities can also be found separately in non-living matter. A magnet retains a memory of an external magnetic field. A crystal grows by incorporating material from its surroundings. Lightning reproduces branching patterns as it propagates through the atmosphere. But when all 3 abilities appear together, something new emerges. Life. Life is not a substance. It is a process. Your body began as a single cell. From that cell emerged trillions of others, organized into tissues, organs, and systems. Even today, we could not recreate that entire process from scratch. The number of coordinated steps is simply too great. But if we step back from the complexity of a complete organism and focus on the simplest action performed by living matter, the copying of a single molecule, the physics becomes far more manageable. And that physics can already be recreated in the laboratory. Let's imagine a molecule that functions like a mold, for making copies of itself. It captures the necessary molecular building blocks from a solution, presses them together, and they bond. The result is an exact copy of the original molecule. That copy, then, does the same thing. This is what the simplest possible act of life looks like without cells, DNA, or any of the familiar machinery of biology. Scientists have been reproducing processes like this in laboratories for decades. In one series of experiments, researchers placed long molecular filaments into a solution and then broke them apart using ultrasound. Each fragment became a new growth point. It attracted building blocks from the surrounding solution and extended itself until it became a complete filament once again. The more filaments that were broken, the more growth points appeared. The process accelerated. The total mass of filaments increased like an avalanche. In 2002, chemists created an artificial RNA molecule capable of doing something similar. The molecule found two short RNA fragments floating in solution and joined them together. The result was an exact copy of itself. That copy then produced another and another. The number of molecules doubled repeatedly. Scientists had effectively created exponential growth in a test tube, a process strikingly similar to the growth of bacterial populations. Perhaps the most unsettling example comes from prions, the infectious proteins responsible for mad cow disease and related disorders in humans. A prion is not a virus or a bacterium. It is simply a normal protein that is folded into the wrong shape. Yet, it behaves almost like a molecular zombie. When it encounters a healthy version of the same protein, it forces that protein to adopt the same abnormal structure. As a result, defective proteins rapidly spread through the cell. Of course, neither RNA molecules nor prions are alive. They are merely complex arrangements of atoms. But they reveal one of nature's most important principles: positive feedback. Positive feedback is a process that amplifies itself. The same principle drives avalanches down mountainsides. It powers chain reactions in nuclear materials. It allows a single infected person in a crowded shopping center to trigger an epidemic. A small change reinforces itself, grows larger, and continues expanding until it reaches the limits imposed by its environment. But there is a price. Every positive feedback loop requires a continuous supply of energy from outside the system. Remove the fuel, and the process stops. Try to assemble a human body atom by atom, molecule by molecule, according to a detailed blueprint, and the task becomes essentially impossible. Nature does not build organisms that way. Instead, it establishes simple feedback loops within environments through which energy is flowing. Those loops accelerate themselves, copy themselves, and gradually become more complex. If you assemble atoms at random, you will not get a living organism. Imagine a disturbing but revealing experiment. Take a frog and put it into a blender. Every atom remains, every chemical element remains. Nothing has disappeared. And yet, that mass of matter will never jump again, never catch flies, and never behave like a frog. To understand why, physics relies on two fundamental concepts. The first is aggregation. Every object has what physicists call a microstate. A microstate is the exact position and velocity of every particle in the system at a given moment. To describe something as simple as a snowflake at this level, you would need an astronomical amount of information, quintillions upon quintillions of numbers. Measuring all of them simultaneously is impossible. That is why both our senses and our scientific instruments usually view the world from a distance. We see a glass of water, not the chaotic motion of the atoms inside it. We compress an unimaginable amount of microscopic information into a few convenient labels. As I discussed in more detail in a previous video, those labels do not have to be physical quantities like temperature or pressure. They can also be biological categories. We can take an enormous collection of carbon, oxygen, hydrogen, and nitrogen atoms and label the entire arrangement frog. From the perspective of physics, there are an astonishing number of possible ways to arrange those atoms. A tiny fraction of those arrangements produce a healthy, living frog. Some produce a sick frog. Most produce nothing recognizable at all. Once again, physics itself does not contain the word life. Human beings invent the categories and draw the boundaries. But once we draw those boundaries, physics can study the transitions between them. The second concept is entropy. Entropy explains why a frog never reassembles itself after passing through a blender. At its core, entropy is simply the number of microscopic arrangements that correspond to the same large-scale outcome. Consider a simpler example. Why does the air in a room never spontaneously gather in one corner? Nothing physically prevents the molecules from moving there. There is plenty of space available. The reason is statistical. There are unimaginably more ways for air molecules to spread throughout the room than there are ways for them all to occupy a single corner. Forcing every molecule into one corner corresponds to a tiny number of possible microstates. Allowing them to spread everywhere corresponds to an enormous number. That is why a uniformly distributed gas has higher entropy. In a chaotic system, all microstates are treated as equally likely. The overwhelmingly more numerous states dominate. Exactly the same thing happens to our frog. The blender randomizes the positions of its molecules. Once that happens, there are astronomically more ways for those atoms to exist as a disordered mixture than there are ways for them to reassemble into a living animal. Chaos wins simply because it has more available configurations. But chaos does not always dominate. Consider a glass of water freezing on a cold winter day. The crystal lattice of ice is a highly ordered structure. Its entropy is lower than that of liquid water, where molecules move freely in a much more chaotic state. So why does order suddenly emerge? Why does the system become more organized instead of less? The answer lies in electrical forces. Water molecules carry tiny electrical charges across their surfaces. Positive regions are attracted to negative ones. Because of this, water molecules can lock together like pieces of a construction set. When the temperature is high, thermal motion constantly breaks these connections, causing the molecules to separate. But as the temperature falls, the molecules remain attached for longer and longer periods. Order begins to prevail over chaos. Physicists often visualize this process as a ball moving across a mountainous landscape. The peaks represent states of high energy, while the valleys represent states of low energy. The natural forces acting within a system always tend to push the ball downhill. At absolute zero, the ball simply settles into the deepest valley and remains there. But at higher temperatures, thermal energy continuously kicks the ball. It climbs the slopes, falls back down, and climbs again. When temperature remains constant, the system reaches what is known as thermal equilibrium. The behavior of the particles is then described by the famous Boltzmann distribution. The probability of finding a system in a particular state decreases exponentially as the energy of that state increases. In simple terms, the higher the hill, the less likely the ball is to be found there. But as the temperature rises, the kicks become stronger, and the ball can explore much larger regions of the landscape. This endless struggle between order and chaos can be captured by a single physical principle. Nature tends to minimize its free energy. For a state to be highly probable, it must either possess very low energy or very high entropy. Temperature determines which side gains the advantage. As temperature decreases, the influence of entropy weakens. Internal energy begins to dominate. The result is that a chaotic liquid can spontaneously organize itself into an ordered crystal. Physicists call this process, equilibrium, self-assembly. If a collection of components contains even one particularly favorable, low-energy way of fitting together, those components will naturally assemble themselves into that arrangement. This is how dissolved salt ions organize themselves into crystals. This is how water molecules arrange themselves into the intricate hexagonal structures of snowflakes. This is how lipids spontaneously form the double-layer membrane surrounding every living cell without enzymes, without guidance, and without any external architect. Pure thermodynamics is enough. Today, scientists are exploiting this principle at the forefront of nanotechnology. One of the most remarkable examples is DNA origami. Researchers can design strands of DNA that fold themselves into complex nanoscale structures, assembling automatically into predetermined shapes. Molecules find their places on their own thanks to precise attractive forces. Nature learned long ago how to encode these forces, and that ability now allows scientists to create remarkable, almost lifelike materials in the laboratory. Now, let us return to our frog in the blender. In that chaotic mixture, all atomic arrangements are effectively random. But imagine sealing those remains inside a perfectly closed container and leaving them at a constant temperature for trillions of years. Could the atoms eventually reassemble themselves into a living frog? Common sense says no. The frog would simply decay. Yet, from the perspective of physics, this question conceals a profound mystery. The main building blocks of life, proteins, are capable of assembling themselves. This was demonstrated by the scientist Christian Anfinsen. A chain of amino acids can spontaneously fold into its correct three-dimensional shape. It simply moves downhill along an energy landscape, much like water freezing into ice. So why can't an entire frog come back to life? There are two fundamental reasons. The first is the energy barrier. Ice is stable because it occupies a low-energy state at the bottom of an energy valley. Living systems are different. Life resembles a whirlpool in a river. It maintains its structure only because energy continuously flows through it. Shut off that flow and the whirlpool disappears. According to the laws of thermodynamics, maintaining such a state requires a constant throughput of energy. In complete equilibrium, the probability of matter spontaneously organizing itself into a living system becomes vanishingly small. The second reason is connected to what physicist Jeremy England describes as the extraordinary rarity of useful configurations. Consider proteins. Nature uses 20 different amino acids as building blocks. A typical protein may contain around 200 of them. The number of possible sequences is therefore immense, 20 to the power of 200. An overwhelming majority of those sequences do nothing useful. They will not fold correctly. They will not catalyze reactions. They will not perform biological functions. Functional proteins are extraordinarily rare structures. They are among the most sophisticated products of molecular evolution. Take myosin as an example. Myosin functions as a genuine molecular motor. It consumes chemical fuel and walks along the internal scaffolding of the cell. But alter just a few amino acids, and the motor stops working. Ordinary equilibrium physics cares only about energy. It does not care about function. A rock resting at the bottom of a valley is perfectly acceptable to physics. A functioning molecular machine is something else entirely. For non-living matter to become living matter, different conditions are required. Heat alone is not enough. A directed flow of energy must pass through the system. Only when energy begins flowing through matter do entirely new possibilities emerge. Everything alive moves. Birds fly, flowers bloom, bacteria grow and divide. But movement alone is not unique to life. A stone rolls downhill. A yo-yo spins on a string. There is a fundamental symmetry in the mechanics of the non-living world. A stone can be kicked back to the top of the hill. A yo-yo spins just as easily in one direction as in the other. But life is different. A tree grows from a seed, yet it never turns back into one. People grow older, but no one grows younger. Why does the dynamics of life seem to move in only one direction? The answer lies in energy. In physics, energy is a way of keeping track of motion. Isaac Newton built his mechanics around the concept of force. Most of the fundamental forces in nature are conservative, meaning that they can fully return the motion that was previously stored. Imagine throwing a ball straight into the air at the highest point of its flight. It pauses for an instant. All of its kinetic energy has been converted into gravitational potential energy. Then it falls back down and regains the same speed. On the moon, where there is no atmosphere, this motion is perfectly symmetrical. If you played a video of the throw backward, nothing would look strange. Newton's equations are completely symmetric in time. What can happen forward can also happen in reverse. But on Earth, that symmetry is broken. As the ball falls, it loses energy through collisions with air molecules. Its motion is fragmented into billions of tiny, chaotic movements. Physicists call this heat. In principle, Newton's equations allow the reverse process. The air molecules could stop resisting the ball and instead push it upward together. The laws of physics do not forbid such an event. But in reality, air resistance always slows the ball down. Molecules never coordinate their motion to help it. Why not? The answer comes from probability. Imagine 100 people standing tightly packed in the center of a town square. Each person begins taking random steps in random directions. After an hour, the compact group will have spread out into a scattered crowd. For everyone to gather back at the center, each person would have to randomly choose the correct steps toward the same point at exactly the same time. That is not impossible. It is simply extraordinarily unlikely. Air molecules behave in exactly the same way. They spread out, not because they are forbidden from regrouping, but because dispersal is overwhelmingly more probable than coordination. Energy behaves the same way. It dissipates. When a ball collides with the atmosphere, its energy spreads outward in every direction. To reverse that process, trillions upon trillions of molecules would have to coordinate their motion and strike the ball in the same direction at the same moment. The odds are unimaginably small. It would be like flipping a coin 1 trillion times and getting heads every single time, and hoping to get heads 1 trillion times in a row. You could wait longer than the age of the universe, and it still would not happen. Randomness creates disorder. And it is this disorder that breaks the symmetry of time and makes the living world move in only one direction. Remarkably, we can mathematically measure exactly how different a movie played forward is from the same movie played backward. In 1999, the young Berkeley chemist Gavin Crooks derived a beautiful equation now known as the Crooks fluctuation theorem. It links the arrow of time to the heat produced during a process. The meaning of the equation is surprisingly intuitive. Every violation of time symmetry has a thermodynamic cost. If a process releases heat, then the forward version of the movie becomes exponentially more probable than the reversed one. The heat has dispersed into the environment. The chance that all of that energy will spontaneously gather itself back together approaches zero. Nature is filled with processes that break time symmetry. The heart beats and pumps blood in only one direction. Rain falls onto mountains and flows toward the sea, but it never runs uphill and gathers itself back into clouds. So where does this directionality come from? The answer is simple. From an external source of energy. The sun evaporates water from the oceans. Winds transport the vapor through the atmosphere. Gravity pulls the rain back to Earth. The heart pumps blood because muscle cells consume ATP. And ATP is continually regenerated using energy extracted from food. Somewhere in the system, there is always a source of fuel, a stream of low-entropy energy entering the process, being transformed, and ultimately leaving as heat. Without that flow, nothing happens. Physicists describe such situations as nonequilibrium steady states. A living cell is an extraordinarily complex network of chemical cycles operating under precisely these conditions. If we recorded a movie of molecules at thermal equilibrium, we would not be able to tell whether it was being played forward or backward. The molecules would simply move randomly in all directions. But the moment a directed flow of energy passes through the system, a preferred direction of time appears. And with it, the possibility of life. Imagine a child pulling a wooden duck on a string. The toy moves only as long as the child keeps pulling. The moment the child lets go, the duck quickly comes to a stop, having converted its motion into frictional heat. Living systems are organized in much the same way. Their string is chemical work, and it is pulled not by a child, but by a continuous supply of fuel. Stop eating, and within a matter of weeks, the body shuts down. This is what physicists call a nonequilibrium steady state. A process that appears stable, yet exists only because energy is constantly flowing through it. But why do living systems need fuel at all? Why can't they simply exist without it? The most obvious answer is growth. Trees absorb sunlight and use that energy to build trunks, branches, and roots. Energy becomes stored in the structure itself. But fuel remains necessary even after growth has stopped. It is required for maintenance, repair, and, most importantly, self-replication, the ability to create copies of oneself. Self-replication leads to exponential growth. It is an ordered, directed process, and every such process has a thermodynamic price. It generates heat. This simple fact opens an unexpected perspective on evolution itself. Consider DNA and RNA. RNA is far less stable in water than DNA. It breaks apart thousands of times more quickly. At first glance, this seems like a serious disadvantage. But from a dissipative perspective, instability can sometimes be beneficial. Less stable molecules renew themselves more rapidly, process energy more actively, and under the conditions of the early Earth, this may have provided an advantage. In a sense, RNA lived faster than DNA. According to current theories, RNA was likely the first major molecule of life, while DNA emerged later as a more stable system for storing information over long periods of time. Any rapid change that locks a system into a new state requires work, and work produces heat. Physicists often represent the possible states of a system as a landscape where height corresponds to potential energy. Without an external energy source, a molecular ball simply wanders across this landscape, rolling from valley to valley under the influence of random thermal fluctuations. If two valleys sit at the same height, moving forward is just as likely as moving backward. The amount of heat absorbed equals the amount released. Introduce an external source of energy, however, and the landscape changes. Now, imagine that an escalator appears on the hillside. External work carries the molecular ball rapidly upward. It crosses the peak, falls into a new valley, and releases excess energy as heat. Without that external push, the transition would never occur. This is how a constant flow of energy gives rise to the arrow of time, and it is how ordinary matter acquires the ability to behave like living matter. Living organisms perform this trick continuously at every scale. Inside cells, specialized molecular machines known as chaperone proteins consume chemical fuel in order to force other proteins into the correct shapes. Left on their own, many of those proteins would never find the structures they need to function properly. And the same principle extends far beyond individual molecules. It operates at the scale of the entire organism. Every gesture of the hand, every step, every muscle contraction is a directed motion that requires energy and ultimately releases heat. That is why our bodies constantly generate heat from within and must continuously cool themselves through breathing, sweating, and thermal radiation. The same is true of the brain. Neurons communicate through electrical impulses, and in the process, they produce a significant amount of heat. Although the brain accounts for only about 2% of body mass, it consumes roughly 20% of the body's total energy budget. It is an extraordinarily expensive organ, and much of that cost is paid for its ability to store, process, and manipulate information through time. Memory, learning, and thought all break time symmetry. They leave traces of the past that influence the future, and according to physics, that always comes with a thermodynamic price. This brings us to one of the most surprising ideas to emerge over the last half-century. Information processing is a physical process. And like any physical process, it has a measurable thermodynamic cost. The connection between information and physics was first clarified by Rolf Landauer. He asked a seemingly simple question: how much energy must a computer expend to erase a single bit of information? To erase a bit, a physical system must effectively switch on the escalator. It must push a particle from one state into another, and, in doing so, release energy as heat. Landauer proved that there is a fundamental lower limit to this cost. No matter how advanced a technology becomes, information cannot be erased for free. The limit is built into the laws of nature themselves. For decades, Landauer's principle remained a theoretical prediction. Then, in 2012, physicists tested it experimentally using a tiny colloidal particle trapped inside a double-well potential. When researchers forced the particle into a specific state, the amount of heat released matched Landauer's prediction with remarkable precision. The connection between information and thermodynamics turned out to be real. Not philosophical, not metaphorical, a measurable fact of nature. Every act of computation, every recognition of a pattern, every memory formed, and every thought processed requires energy to be dissipated as heat. In that sense, we are thermal machines running information processing systems. And the reverse is equally true. Information processing cannot exist without energy flowing through a physical system. Another remarkable property of life is its ability to repair itself. A cut on your skin gradually closes. Damaged tissue rebuilds itself. From the outside, it almost looks like a miracle. But physicists view this process differently. A hole in a frozen lake also repairs itself as ice gradually forms across its surface. Yet, this is passive repair. Water molecules simply slide downhill along the energy landscape, release excess heat into the cold air, and settle into a crystalline lattice. No one guides them. They move toward the state with the lowest free energy. Many biological structures assemble themselves in much the same way. DNA strands find their complementary partners and bind together. Viral shells self-assemble from individual protein components, like three-dimensional puzzles in which every piece fits only one location. Here, nature relies on equilibrium self-assembly, the same principle that creates salt, crystals, and snowflakes. But passive self-assembly has a fundamental limitation. Structures that sit at the bottom of an energy valley are stable precisely because they resist change. They are inert. They do not respond dynamically to shifts in their environment. Life requires something very different. It requires structures that are both stable and adaptable. And such structures do not emerge spontaneously from equilibrium. Living repair operates according to a completely different principle. A crystal is frozen in place. A living organism is not. Life is a continuous cycle of damage and repair, destruction and reconstruction. An ice crystal rests comfortably at the bottom of an energy valley. A living organism is more like a runner sprinting up a downward-moving escalator. To remain where it is, it must constantly expend effort. Inside every cell, thermal chaos is at work every second. Proteins misfold, DNA strands are damaged, cell membranes oxidize. Destruction is not an occasional event. It is a permanent feature of life. If you place a green plant in complete darkness, it will eventually die. Physical death is the final surrender to thermal equilibrium. For a while, the plant can resist. But without a source of energy, disorder gradually takes over. The moment light returns, everything changes. The plant captures packets of energy from incoming photons and restarts its internal machinery. Sunlight becomes the fuel for millions of microscopic repair operations occurring every second. Molecular chaperones refold damaged proteins. Cellular systems restore depleted chemical stores. This invisible maintenance work is what keeps matter alive. The healing of a major wound is simply a dramatic manifestation of a process that is already happening continuously. But why did life choose this path of endless energy consumption instead of the quiet stability of a crystal? Because a crystal is stable precisely because it cannot change. Life demands something far more difficult: stability combined with flexibility. A living system must preserve its structure while remaining capable of rapid adaptation to a changing environment. Cells solve this problem using molecules such as actin. Actin filaments weave themselves into a complex internal network that serves simultaneously as a skeleton and an engine. They help cells maintain their shape, move through their environment, and reorganize themselves when necessary. The secret is that every actin filament continuously consumes chemical fuel. The structure is constantly being dismantled and rebuilt in real time. Physics leaves no room for compromise here. You cannot be both highly stable and highly adaptable without continuously dissipating energy. Of course, energy can flow through non-living matter as well. Wind blowing across, sand creates dunes. If the wind stops, the dunes do not vanish immediately. But over time, countless random collisions with air molecules gradually erase the structure. Does that make a sand dune alive? No. Consider another example. Place a marble inside a glass and spin the glass rapidly. The marble climbs the wall and remains suspended there as long as the motion continues. Stop spinning, and the marble falls back down. At first glance, this seems similar to self-maintenance. But the marble knows nothing about its state. It performs no computation, it stores no information, it simply responds to external forces. Life is fundamentally different. A living system maintains thousands of interacting structures simultaneously, each carrying out a specific function while energy flows continuously through the whole network. But sometimes, even non-living nature can surprise us. As water carves its way through landscapes, it creates river networks of astonishing beauty. These branching patterns look almost organic. And even more, remarkably, they obey precise mathematical rules. Researchers at MIT studied how river systems form and discovered something unexpected. River channels tend to branch at a very specific angle, about 72 degrees on average. This angle appears to optimize the balance of water flow and maximize access to groundwater sources. Even more astonishing, if such a river network is filled in with sediment, the flow of water will eventually carve out nearly the same pattern again. In other words, a non-living system can recover its own structure. Interestingly, similar branching mathematics appears elsewhere in nature. The same geometric patterns can be found in networks of neurons within the brain and in the sprawling filaments of fungal mycelium. This is not a coincidence. The same underlying physics can generate similar forms in both living and non-living systems. River channels develop characteristic branching angles because water and soil interact according to simple physical principles. But every physical interaction is, at its core, an exchange of energy. One billiard ball strikes another and transfers momentum. A bullet impacts a target and converts its kinetic energy into heat. In simple situations, the flow of energy is easy to follow. But in nature, the story becomes far more interesting. As we are about to see, matter does not absorb energy indiscriminately. It absorbs it selectively. The transfer of energy often depends on a perfect match. Throw a log into a fire and it bursts into flames almost immediately. Leave the same log lying on cold ground and nothing happens. A child on a swing rises higher and higher only when the timing of the movements is just right. Push a little too early or a little too late, and the swing begins to slow down instead. Even a bacterium requires a very specific DNA sequence in order to metabolize sugar. Without that microscopic configuration, it cannot process a single sugar molecule. Matter absorbs energy selectively, and that selectivity depends on its current state. The secret lies in frequency. An empty bottle resonates at one pitch, while a bottle filled with water resonates at another. A stretched rubber band produces its own characteristic tone. Every object possesses natural frequencies of vibration determined by its size, shape, and material properties. This principle also works in reverse. Objects do not merely produce vibrations, they absorb them as well. An opera singer can shatter a wine glass with her voice. The secret is not simply loudness. The glass breaks only when the singer matches its natural frequency. This phenomenon is known as resonance. When the glass bends slightly to one side, the sound wave pushes it further in the same direction. When it bends back, the wave reinforces that motion as well. With every tiny oscillation, more energy accumulates within the structure. Eventually, the deformation becomes so large that the glass can no longer withstand the stress. It shatters. What makes resonance especially interesting is that it can be changed, and you do not necessarily have to alter the sound. You can alter the object itself. Change the shape of the glass, and you change the frequencies it absorbs most efficiently. The same matter, arranged in a different configuration, interacts differently with the energy around it. And absorbed energy has remarkable power. It can rearrange matter itself. It can make atoms move, reorganize, and adopt entirely new structures. The crucial point is this: matter tends to move towards states that provide better access to available energy sources. But how can we determine which structures are best at absorbing energy from their environment? The answer begins with mechanical resonance. Any large collection of particles possesses its own set of natural frequencies. In a sense, these frequencies form a unique barcode for a particular arrangement of matter. Every structure is capable of absorbing enormous amounts of energy, but only if the external force matches its internal rhythm. If the surrounding environment is, so to speak, singing at a particular frequency, then the appropriate structure responds through resonance. And at that moment, the energy escalator switches on. The system is pushed out of its old valley and into a new configuration. But that new configuration possesses an entirely different set of natural frequencies. Something similar happens in biology. Living organisms change through mutation and selection. A useful mutation appears. Natural selection favors it. And over thousands of generations, the entire population may be transformed. Once that transition occurs, there is usually no simple path back. Evolution moves forward. Non-living matter can undergo an analogous process. Every structure is tuned to particular frequencies. If the energy arriving from the environment matches those frequencies, the structure begins absorbing energy and transforming itself. The new structure that emerges then possesses a different set of resonant properties. Returning to the original state becomes statistically unlikely. In this sense, matter can be said to evolve through a sequence of resonant transitions. Consider the example of the singing glass. It encounters a perfect resonance, begins vibrating more and more intensely, and eventually shatters. What happens after that? The story ends. The fragments no longer resonate with the same song. The system has reached a state where it can no longer efficiently absorb energy from that particular source. It becomes trapped, and yet the shattered glass still contains information. The positions of the fragments are physical traces of the event that created them. They are scars left behind by a past burst of energy. Like archaeologists reconstructing an ancient catastrophe from scattered remains, we can infer the history of the event from the final arrangement. The ability of matter to absorb and transmit energy depends on its internal architecture. A beautiful example comes from what physicists call Rayleigh-Bennet cells. When a layer of liquid is heated from below, orderly convection cells spontaneously emerge. These circulating vortices transport heat far more efficiently than random molecular motion alone. The same principle appears in a blacksmith's forge. Cold metal resists the hammer. Its atoms occupy relatively stable positions deep within an energy landscape. But heat the metal until it glows red, and those atoms gain access to a much larger range of states. The metal becomes far more willing to absorb the energy of each blow and transform its shape. This process, however, is not especially selective. Any sufficiently strong impact can deform hot metal. Resonance is different. A wine glass shattering from a singer's voice requires an extraordinarily precise match between external energy and internal structure. Physics offers many examples of how the architecture of matter determines the way energy is absorbed. Form matters. The specific arrangement of matter determines what kinds of energy it can capture, process, and respond to. And where does this entire chain of ideas ultimately lead? It leads to the emergence of complex life, because any piece of matter through which energy flows can, in principle, be evaluated by a single question: how effectively does it absorb, process, and dissipate that energy? And this leads to a strange question. If life is ultimately a particularly sophisticated way of channeling energy through matter, then is there such a thing as an ultimate form of life? A form that extracts the maximum possible utility from its environment? Over the past half-century, several prominent physicists have taken this question seriously, and many of them arrived at surprisingly similar conclusions. In 1979, physicist Freeman Dyson published a paper with an almost theological title, Time Without End: Physics and Biology in an Open Universe. He asked a fascinating question. What happens to intelligent life when the universe cools to temperatures approaching absolute zero? The intuitive answer seems obvious. Life dies out. Dyson disagreed. He argued that if energy is managed efficiently enough, life could continue for an extraordinarily long time, perhaps, effectively, forever. The idea behind what is now known as Dyson's eternal intelligence is based on Landauer's principle, which we discussed earlier. Every bit of information processing requires a minimum amount of energy and generates a corresponding amount of heat. The colder the environment, the cheaper each computation becomes. Dyson realized that a sufficiently advanced civilization could adapt to this reality. As the universe cooled, intelligent beings could gradually slow their metabolism and thought processes. They would think more slowly, act more slowly, and process information more slowly, but they would also consume less and less energy. In principle, this strategy would allow a finite supply of energy to support an unlimited amount of subjective experience. Put differently, finite fuel could produce infinite subjective time. Dyson did not claim that such beings would necessarily exist. He simply demonstrated that the laws of physics did not obviously forbid the possibility. More importantly, he showed that the idea could be analyzed quantitatively. Later, discoveries complicated the picture. The accelerating expansion of the universe revealed that distant regions of space eventually become causally disconnected from one another. This creates serious challenges for Dyson's original scenario. But the underlying idea survived. The notion that life might evolve toward forms perfectly adapted to the energy flows available in the universe remained deeply influential. 2 decades later, in 2000, MIT physicist and engineer Seth Lloyd explored a related question in a paper published in Nature with an equally provocative title, Ultimate Physical Limits to Computation. Lloyd asked something even more concrete. What is the most powerful computer that could possibly be built from 1 kilogram of matter? He took the known laws of thermodynamics, quantum mechanics, and general relativity and pushed them to their absolute limits. The result was astonishing. If every physical resource available within 1 kilogram of matter were used with maximum efficiency, the resulting device could store roughly 10 to the power of 31 bits of information and perform about 10 to the power of 51 logical operations every second. Those numbers are almost impossible to visualize. For comparison, a modern smartphone stores roughly 10 to the power of 13 bits of information and performs around 10 to the power of 10 operations per second. Lloyd's ultimate computer would possess hundreds of trillions of billions of times more memory and would perform calculations at speeds that dwarf anything humanity has ever built. Physically, such a machine would resemble an extremely dense plasma heated to billions of degrees, a tiny fragment of the Big Bang confined to the size of a laptop. And if that same kilogram of matter were compressed even further down to its Schwarzschild radius, forming a black hole, something remarkable would happen. The maximum rate of computation would remain essentially the same, but communication between different parts of the system would become extraordinarily efficient. Lloyd showed that, in a very real sense, the fastest possible computer allowed by the laws of physics is a black hole of the appropriate size. This might sound like nothing more than a curious, theoretical exercise. Except for one unsettling fact. Life itself is an information processing system. A cell continuously reads genetic instructions and makes decisions based on them. The brain transforms sensory input into action by consuming energy and dissipating heat. If evolution truly drives matter toward increasingly efficient ways of channeling energy, then over sufficiently long timescales, it may also drive matter toward greater computational efficiency. In that sense, the ultimate form of life and the ultimate form of computation may not be separate things at all. Lloyd is careful not to present this as a prediction. These are upper limits, not prophecies. But the boundary he identified reveals something profound. There is a fundamental physical ceiling on how effectively matter can process information and channel energy. Viewed from this perspective, life on Earth is merely one solution among many possible solutions. Carbon-based chemistry operating in liquid water happens to be a highly effective strategy under the energy conditions available on our planet. But there is no reason to assume it is the only strategy. On worlds dominated by hydrothermal vents, methane seas, subsurface oceans, or entirely different energy sources, the optimal form of life could look radically different. The underlying principle would remain the same, while the physical implementation would change. And on truly cosmic timescales, the most efficient form of life may not be biological in the traditional sense at all. It may be a computational structure designed to push matter as close as possible to the fundamental limits allowed by physics. If life is genuinely drawn toward those limits, then our own evolutionary history is not merely a sequence of accidents. It is a trajectory. Each major transition increases the density with which energy, information, and computation flow through matter. The first self-replicating molecules were one step. Cells were another. Multicellular organisms followed. Then nervous systems. Then brains. Then language. Then science. At every stage, matter discovered new ways to store information, process information, and exploit energy more efficiently than before. Seen from this perspective, evolution resembles a long climb toward increasingly sophisticated forms of organization. And where are we on that journey? Somewhere in the middle. Complex enough to ask these questions. Complex enough to understand the laws that shaped us, but still unimaginably far from the limits that physics allows.